And we do always like to do a land acknowledgement. It's important. And this is
Yes.
Indigenous History Month, I believe. So I feel that this is extra important. I think that's it.
Excellent.
I will
So,
put our open notes in the chat, and that way anyone can add their name and make notes in the document. So, okay, now it's your turn, Judith.
Thank you, Alex. So, welcome everybody. As we say in the Wendake language, Kwe. Yes, today in this BINA AIMM meeting, Automated Image Management and Metadata Annotation, we have the pleasure of welcoming Anthony Hasmar from the NIST, the American National Institute of Standards and Technology. And Asmar is going to, Anthony is going to present us today the importance of metadata beyond the microscope in the lab. So thank you very much, Asmar. We are very excited to welcome you in this group today. I think you are still muted.
Sorry, trying to share my screen. So can everyone see my presentation, hear me fine?
Looking great, thanks.
Okay, all right, thank you. So yes, I'm Anthony Asmar. I work at NIST. NIST is a National Institute of whoa, sorry, wrong presentation. Here we go. Sorry, I've given a lot of talks in the last week. So I work at NIST, and NIST basically is the U.S. National Measurement Laboratory. And really what I want to focus on is the important aspects about NIST is that we're non-regulatory, but we do partner a lot with regulatory agencies. And we also partner a lot with industry. And really it's just, we partner with them in order to kind of support measurement science itself. And so when you think about NIST, a lot of people just think standards. Standards is only one aspect of what we work on. We kind of really think about the measurement science and the infrastructure for that, which includes like protocol development tools. There are reference materials and reference data, but then what I really focus on is like new measurement technologies and kind of thinking about where everything is going to, we have to understand it if we want to start building standards for it. So, unless we kind of design and build it ourselves, I mean, with input from outside as well and collaborators, we can't really move towards developing standards in that sense. So that's why I kind of work on these new measurement technologies. So I'm kind of going to dive right in. So, I mean, I think BINA, most of the group here, the people in this room are mainly, mainly focused on bioimaging, but probably bioimaging as a tool to kind of learn something new in a biological process. Kind of as a, but I think when we think about it from a different aspect, and what I really focus on is from a manufacturing process. And so when you think about it from manufacturing, it's very different than biological research because it's not just that you're trying to learn something new or like trying to explore and get a novel measurement. Instead, what it is, it's really the science of making the same thing, so you want to make sure it's repeatable. And this is why also, having standards, having good protocols, all these things, benchmarking will be really important because you always want to make sure that the measurement tools you're using are also repeatable. And so you're getting the same proper input so that you can actually measure your process properly. And so I'm going to talk about just quickly Industry 4.0. I know it sounds like kind of almost a marketing gimmick in a way, but it really is a real thought process of kind of characterizing the different industrial revolutions that have occurred over time. And so we can say back in the 70s, 80s, there was the Industry 3.0 kind of started. And that's when we started having computer communication, networking of devices, and then also enabling what they say continuous manufacturing, where we have these analytical technologies that kind of give input and we can kind of keep moving through the process without having to stop take a measurement ourselves, like remove the samples, do things. And so this has kind of revolutionized how drug manufacturing. So a lot of stuff is kind of in the perspective of, let's say, car manufacturing, other types of material manufacturing, drug manufacturing. But we're really starting to move now into focusing on biological manufacturing and biomanufacturing. And I mean, that's only kind of happened more recently with a lot of, let's say CAR T therapy or, stem cell therapy, which is what I focus on. But right now we've moved kind of from Industry 3 to Industry 4.0. And so Industry 4.0, and this is from like a 2021 paper from FDA on, talking about drug manufacturing. But really it's saying that the new kind of way of thinking is that you want to integrate multiple systems. You're going to digitize data and work with very large data. And I mean, this is kind of, and then start incorporating artificial intelligence. And then having that, you can develop actionable information that allows for autonomous manufacturing. And so this is where the system itself can continue to control the whole manufacturing process without really needing someone to intervene all the time or be monitoring all the time. So I'll kind of go a little bit into like what a biomanufacturing process looks like. So let's say we're doing something on a microscope. We're going to just run a single assay. You can kind of think about it the same way in a biomanufacturing process, that we're doing a process, like one thing, and then we can characterize it or kind of group it as a unit operation. And between each unit operation, we usually have a measurement process that occurs, this analytical process. And then we usually get some sort of, into some sort of data model. And then we can decide if we want to move on to the next step. And then we have, unit operation 1 and 2 and 3, and then before we get to a final product. But one thing I wanted to say is that, each of these operations has multiple steps, different protocols, and not just one measurement, but many measurements. And this also generates a lot of metadata, or you need a lot of metadata. And that metadata is important because it kind of gives context to what's going on, the outputs that you get, and it helps encode provenance. And so this is a term that's used a lot in NIST, and then we think about that provenance as really having that chain, like the chain of quality, the chain of control, and that traceability. And so this is critical in manufacturing. Doesn't matter what, like what area of manufacturing, having that provenance is important. And then that's also important for regulatory process, because you have to make sure you have control of the product as you're developing it, or, the biological material as you're developing it throughout the whole, biomanufacturing process. And so now what we're kind of moving into is that instead of just taking this measurement, in between each unit operation or a specific step, we're really kind of moving towards continuous monitoring. And this is where I think, and this is what I've kind of had a lot of discussions with industry, is that this continuous monitoring, especially having direct monitoring of, let's say, when you're growing cells, is that bioimaging itself is critical for this. And I think that's actually one of a lot of the discussions that I hear at a lot of these manufacturing conferences, that they're moving towards biomanufacturing to be as a tool, as a process analytical technology tool to be used for continuous monitoring, especially when you can use it without destroying the cells or, sacrificing a sample. And so why is provenance important? This is what they call a fishbone diagram or Ishikawa plot. And so this is not comprehensive. Let me just put it that way. This is actually just kind of looking at some of the things that are in in like one assay, a single assay. These are all the things that can go wrong or influence and basically introduce variability in your measurement or the output of the assay. And so we have to be able to kind of understand it, because, we can try and control for everything. We may not be able to control all of it, but at least if we understand where the possible sources of variability are coming from, then we can really understand the measurement that comes out. And so this is really focused about the measurement output that you get. And so we actually build systems to kind of try and, whether we're benchmarking or we do certain controls and everything, to kind of understand all these little pieces that are in this diagram. And so I'm going to move on. This is actually a real iPSC biomanufacturing process. This is for like an autologous therapy for, let's say, neurons. So I was at the International Society for uh, Cell and Gene Therapy, and I was part of I'm more part of a working group there, but I was also part of a panel session. And so me and a lot of the other panel speakers kind of, they're all mainly from industry and from FDA, like kind of regulatory, like consulting and everything. And so we kind of discussed what does a iPSC biomanufacturing process look like? And then we actually compiled also all the, analytical measurements that we might make. And so the way it works is that let's say we take cells from a patient, then we have to reprogram those into iPSCs, into induced pluripotent stem cells. Then we're going to expand, we're going to select which clones that come out of it are the clone that we want. Whether they're differentiated properly or they look like they're differentiating instead of being pluripotent or stem cells. We have to expand them out and grow them out and then freeze down a master bank, characterize that master bank, then, take one vial of that and develop a working bank, then do the differentiation to, let's say, make a neuron or a muscle. There's a lot of different therapies going on, like Parkinson's, Duchenne muscular dystrophy, and all these things. And then you have to actually do a very thorough characterization for the release. And then you would actually also evaluate the clinical efficacy. And so each one of these steps has a lot of different measurements that could be incorporated. And so right now, what I can tell you is that bioimaging is being applied in where the solid lines are. So we have the clone selection, the expansion, and the differentiation. And so one thing I want to say is that, this is talking about imaging not as like a qualitative tool, but using it as a quantitative tool. And so whether it's like texture-based metrics, AI-based, kind of quantification, but basically like a classification to see, what is areas that look like pluripotent stem cells versus differentiated cells, and that's how you can choose which clone you want. There might be other texture metrics that might be important for that. During the expansion, you're checking like confluency as the cells are growing, you can use as a monitoring process. For the differentiation, there's definitely going to be a morphological change when you start differentiating these cells. And so, but you have to have a good way to actually develop that and characterize how you measure it and turn it into a quantitative output. Now, the dotted lines is where I kind of see, and there is some development as well, and I do it myself, is kind of how we can characterize even more steps of the process. And I think that I'll just do a little plug of my research just to kind of give you an idea of what I mean by that, is that, what I work on is actually dynamic continuous monitoring of cells. And so I actually segment every individual iPSC in like a culture, and then I track them over time. And so I think that tracking gives you that temporal data. So we're getting spatial and we're getting temporal data. And I think that spatiotemporal data also gives an extra depth. So it gives you a deeper characterization of the population. But on top of that, when we think about, if we just look at a morphological like texture, it's kind of looking at just the average of everything. We're trying to characterize every individual cell across the population so we can actually get a distribution. And so having that distribution kind of gives that added depth that I was talking about. And so you might be able to use that to actually create a specification of how the cells should behave. We also look at cell cycle time. So I'm monitoring like mitosis events and I'm tracking, doing lineage tracing. And so I know when one cell divides, when does it divide again? And so we can use that to actually measure the cell cycle time. And so this kind of, I think, provides maybe new tools, basically to think about it as a new tool to be able to characterize a culture system and use it for, continuous monitoring. And so if we look at just one of the steps that I was talking about, let's just say the expansion process, how would this be implemented? And so this is where a lot of automation has to come in. We have a cell preparation. We got cells, let's say they're in a dish or they could be in a flask or something, depends on the size. A lot of times when you're working with just patient cells, you might just be working with just 6 well, dishes, 6 well plates. But there's also a lot of metadata that has to be in there, and that's separate from just the imaging metadata. So we've got, whatever the operator, all the information about the person who did this, the technician, there's the plates set up, the protocols, all the reagents also have a whole history of provenance of where the reagents came from, what's their lot, expiration date, as well as any type of documentation that goes with that. Then we have our imaging system, and then let's say we take the image of the cells. Then we have to transfer it to a data storage. And then from there, we have to do maybe, let's say, pre-processing, like I do image stitching and such. And then we're going to do AI processing to maybe segment, but then we're going to do tracking. So that's quantification. But we have to also make sure we keep track of all of those things. So this is kind of getting that data provenance combined with the cell provenance. And then we can get our measurement output afterwards at the end. And so all these measurements then, can be used to either, let's say I want to look at the measurement output and then make a decision. Do I split the cells, or do I change the media, or do I use it, basically make my master bank at that point? Or what a lot of people are doing now, and there's like a shift in that, is that, you can use an AI or a digital twin, and I'll talk about it a little bit later, is that you can make autonomous decisions using that. And so this is kind of what I've been building as well, trying to move towards that autonomous manufacturing and kind of exploring how do you build those models, how do you develop it, how do you validate, and how do you, characterize how well it performs. But I mean, this is only just one thing. This is just monitoring with imaging. But at the same time, there's a lot of other measurements that go in, and they're coming from different sources. So we have to, I was talking about just imaging. And so this is something, when I was part of that panel, we had experts that are on, who do a lot of genetics, karyotyping, whole genome sequencing, other people who do a lot of proteomics, and then also like expression-based panels. And so, there's, when I say like an analytical model, there's like a bunch of data models at each step. And each one of those may have multiple different inputs. So we have to find ways to kind of connect them, bring them together. And that's where I think like metadata really comes into it. But this is why, and also has to be automated, because it's a lot of data to deal with just by hand. So, and this is kind of like what we really think about here is how do we automate these processes? How do we link? How do we design these systems? And so this is kind of like our integrated systems design that we have at NIST. So, I don't know if you've heard a lot, like some people talk a lot about LIMS, which is like laboratory, like information management systems. And so we have whole labs that are just all LIMS capable, every single thing, your centrifuge, your water bath, your incubator, and we actually log all of that data. We take all that telemetry data and then try to put it out as a timeline. But linking all of it is not easy at all. There are some solutions, commercial softwares, that can link certain things and incorporate certain other things. But then let's say you're taking your imaging, how do you incorporate that into it? How do you integrate it? And so really it comes down to, you have the areas that are you're generating data. And so you're not necessarily always doing it in the exact same lab. It might be in a different lab. It might be in a different facility even, that you do some certain measurements depending on what it is. Like if I want to do mass spec, I'm going to have to go over, 3 buildings away, but how do I also link that data back? So it's thinking about how you get the generation of the data, the persistence. So, we have like network storage, but then we also have all the backups as well. We have a local backup, secondary backup, then we have like AWS, Glacier, for like long-term, archiving. And then you have, databases that you need to kind of, in pointers, you have to be able to link everything. And that's where also metadata comes into play, where you have to link multiple different sources of provenance all together. And then you also have to be able to pull it and do all your analysis and quantification. And then ultimately, trying to have maybe a single output or some sort of data gateway to kind of visualize everything like a dashboard. And so that's kind of like the thinking that we do a lot here, is kind of building these large integrated systems. So I was talking about standards. One thing I'll say is right now in ISO 172, there's a proposal for a metadata for image acquisition. This was proposed back in January. I just joined ISO 172, in working group 5, I think, or 3. I forget which one it is. Yeah, but it's basically there is a proposal for a metadata standard for image acquisition. But before we get to there, I also want to think, the reason why I'm talking about this is also that we need to think about it not just from the one single instrument and kind of contained, perspective, but we have to think about there's also going to be a place where, a time where these systems are going to be integrated into larger ones. So we have to also make sure that there's certain levels of compatibility on a larger scale and think about those methods. And so I'm also in ISO 276, which is the biotechnology ISO group. And in working group 5, there is a provenance information model for biological material and data. So the first publication for it was back in 2023. And it kind of used something called open provenance model, which is based on, it's like an implementation of ProvDM, which is a W3C based like provenance model. And right now they're actually working on a document on kind of use cases of it. And so that includes like cell counting, AI and machine learning training, you know, pathology, diagnostics, you know, glass slide preparation, biobanking. But it, you know, it's kind of a nice thought process to show how can you link a lot of data, a lot of different provenance using especially metadata. And so it's just kind of like a modular provenance standard that's being used. And so, you know, it kind of covers, you know, forward and backward linking, forward and reverse connectors. I'm not going to go into it that much because I don't know it that well. I still have to implement it. And I, you know, until I kind of implement it, I don't, you know, can't truly understand, you know, the real limitations of it. But I think there's a lot of groups that, or a lot of communities that are kind of using it in order to use it in like much larger to integrate. It's basically very useful to integrate things on a large scale for provenance, and it's really focused on that. So, and I mean, this is just kind of an example of it. So there was a publication talking about it as like a distributed provenance. And so it's kind of finding ways to, you know, link things. So they kind of bundle like metadata and data and have that provenance and bundling. So they do what they call provenance bundling, and then they're able to kind of pass it on and link them at different steps in the chain. And so it's kind of how do you link things taken from a different laboratory in a different area and like different kinds of metadata or provenance itself, and how do you link them all together? And this is kind of like what the thinking is behind this, using this, the open provenance model. So, move on kind of to talk about, you know, I was talking about, you know, how Industry 4.0, I think the other thing we have to kind of keep into consideration is that there's a lot of AI kind of going on. And so I will say that in the medical devices, so I met with people from FDA even years ago, and they were saying like 50% of all the applications for medical devices were basically all AI based. Um, you know, endoscopes, you know, all these things. And so right now at this point, I think within the last year, they say almost 1,000 AI medical devices have been approved by FDA. Now that's on the medical device side. Now, biomanufacturing and the manufacturing process is still lagging behind, but there's a lot of people working on those tools and trying to develop them. And I can tell you that just from my experience talking with a lot of, you know, companies, you know, a lot of different biotechs, a lot of those AI applications are really focused on image-based systems. So it's all imaging based. And so I think that's something that we have to be, you know, kind of aware of, you know, especially if we're developing any kind of tools. Also, if we want to share data and so on, we have to think about that, you know, there might be, you know, different communities that, you know, we're usually kind of isolated from or not really reaching out to that actually might be a good community to connect with. But really in those, especially when it comes to, let's say, as far as regulations, there's not much written. There's certain guidances and discussions, but they're not very well, there's nothing saying you have to do this, this, and this. You know, they don't ever really say that. But I think things you have to think about is that you have to maintain provenance of your material that you're using. You have to have provenance of the data and how it was treated along the whole way. And if you're using AI, you have to consider the model provenance. Hasn't been modified in any way and so on, and how you validate the data. And then also what are the training that you do as well, and like separate your training data versus your testing data during that process. But you know, those are all critical pieces of information. And so, you know, there's like these two documents I have at the bottom, which are from FDA. And then recently, from ISCT, there was kind of a group, there's like a few different working groups, and they came up about talking about AI, machine learning, and digitization, and how it's kind of what are the use cases that are already out there, in for cell and gene therapy, and then kind of where are things kind of lagging behind. And I think part of that comes down to data sharing and integration. These are the challenges and future directions they talk about. And really, what the biggest thing is that I think, because of proprietary information and things like that and IP, I think a lot of biotechs, a lot of companies are very hesitant to actually publish their data sets. But that's why one of the biggest things that's probably, if you want to make a robust data set or AI model, you kind of need more diverse data sets to kind of feed into and train with. Um, and then, so that's actually it's kind of like, I don't know, catch 22 in a way where it's like, you need more data, but no one wants to share their data. And I think that's actually been a problem in industry. The question is whether there's ways for them to tap into, let's say, some of the more other bioimaging repositories that we have, or kind of make it more aware. It might be also a community to reach out to. So there could be, like I said, some connections there that I think we have to be kind of also aware of, or, kind of think about. Um, I will digital twins. So I don't know if people here know what digital twins are or have ever worked with them, but essentially you can think about it. It's not necessarily always an AI model, but it could be a one single model or a collection of models that kind of is supposed to create a virtual representation or a digital mimic of a physical system. And they use this a lot to kind of, let's say, create a digital. I've seen some really cool ones like where they do digital models of like robotic arms and so on. And so they can actually they can see how the robotic arm is moving on like a computer screen without having to watch it on a video, but they have like the XYZ, like actual like distance, and kind of have a digital simulation of it, of what's going on in real time. But essentially, it can be applied the same way in biomanufacturing, but it's a system that kind of has two-way flow of information. It has inputs and it has outputs, and it kind of can interface with your manufacturing robotic system that allows you to kind of control and can make autonomous decisions. And that's the way that we're trying to work towards. And one example I can say is that, like, here, let's say you're doing an assay and you're growing an assay plate, you follow a protocol. Normally, it's like you seed whatever 100,000 cells in a well, and then you wait, overnight, 16 hours, and then you run the assay. The cells kind of all come together, and let's say they're all confluent, and you're running this assay. The thing is that, you know, because we can't be there, we can't control it, we don't know. We just know that I think they grew enough 16 hours later. But if you have something to monitor it, to characterize it, you can make the decision whether maybe they're already fine at 12 hours, maybe they look better, maybe they need to grow longer. So unless you're there physically present, you have to have something in order to make that decision if you're not there. And so that's kind of when you think about having uh, like finding the optimal conditions without just following a straight protocol, because there's sometimes a little bit of leeway when you say, especially these like long, these long like incubation times, say grow cells for 16 hours. But what if your cells aren't behaving properly? And this is actually one of my experiences, is that when you thaw even the same batches of cells, some of them may take 1 day to recover. Some of them may take 4 days or 7 days or 23 days. So I did a study with 16 different 16 different vials. And so coming frozen at different times with the same cells, and they all recover kind of in different times. So, and normally you get, let's say, cells, and they say their cell cycle time is about 16 hours. Well, if had something wrong with the temperature, or, the media was slightly degraded, or I don't know, it was an older batch, it sat out in the room temperature too long or something, and your fetal growth factor degraded, it's like the cell cycle time might be 20 hours versus 16 hours. And so, that's certain population doublings you're missing. And so this is what I'm talking about when you're doing assays, even especially in an industry setting, you use those assays to characterize like a product, whether it's a drug product or a biological product. And so that actually basically introduces certain variability into your assay. And this is the way that I think about it, is that from a measurement aspect, this is what normally FDA in the past, most people call just inherent variability. It's just like variability that they don't know how to characterize or they haven't really looked into it. And it's really this biological variability that can occur. And so this is why I'm trying to move towards kind of having a more autonomous process or a better monitoring in order to really decide when is the best time to run the assay, or are the cells, or even to kind of characterize the cells or give them like a score saying they kind of fit this level criteria, they fit within this range. And so part of my work building these is that it takes a lot of maybe development and training to do. And so what I've been really trying to look at is how can we do, let's say, rapid AI development. And this comes down to annotation of data, like annotating data. So it's seeing if can we use certain tools like and I mean like biological tools, so like using fluorescent cells or whatever to actually automatically annotate data for us. And what are the limitations versus me hand annotating, thousands of stem cells? And so where does that come in? And this is actually a paper that we had last year that we published, in which what we did was we basically would acquire just one well of a 6-well plate, a 6-well dish, 6-well plate with just stem cells. And they also had an H2B fluorescent tag for GFP. We acquired both of those, 200,000 cells. Take me about 6 to 10 minutes to acquire. It was like 200,000 cells, which was about I think it was like I acquired 100 fields of view, but it was about 56 that I used. It was like 225 million pixels. I don't know why I know that, but it was 214,000 cells that were basically quantified, and we just basically segmented the fluorescence using just basic algorithms, and then we use that to actually train and overlay it with the phase training data. And then from there, trained a 2D UNet for segmentation. And then we've kind of validated using the rest of the data. And so that was actually pretty interesting because we could make a model in like a few hours, like 4 to 5 hours, basically. I mean, other than culturing the cells. So in a day, you can almost have develop basically an AI model to segment cells. And so that was kind of like, how can you implement this in a real environment in a very quick, very rapidly. And we're doing the same for creating basically classifiers for whether the cells are pluripotent versus differentiating. So I've been doing gene editing on stem cells to add pluripotency markers or tag, like basically reporters for their pluripotency. And so this is kind of developing and really focusing on using imaging as a tool for monitoring. So using bioimaging to incorporate into this whole process. So if we say we can characterize what our target cell state that we want, so what is the output that we want of the cells, we use the imaging to basically generate, at least in manufacturing and like we use quality attributes. It just means a measurement output essentially. And so we're doing real time, measurement outputs, and then we can feed that into a digital twin, and the digital twin can then see what do we want to change? Do we need to change media? Do we need to split the cells? Are the cells growing fine? Do they need to go longer? It makes a lot of different decisions. These are what they call process parameters. And then it actually can overlay and control our robotic system that we have. And then, if it changes the media, then we should see a cell response. If it adds more or less, glucose, the cells should respond to that. And so it's kind of this, circular system of where we're measuring and monitoring and continuously going until we reach like the target state that we want. And so that's what my research is mainly focused on here at NIST. And so this is what I work on. Implementing that. So this group is also on automation. I have to think about this a lot, and I spend a lot of time thinking about how to automate. There's not good tools out there to just do that. So this is kind of what I've built now, is that let's say we have our instrument, our microscope. As the data is being generated, it's a lot to process, so I generate a lot of data. And so I do on-the-fly processing. So I actually, as the data is being generated, I actually transfer, I rename, I do folder organization, and I also append metadata. I send it to a local cluster that I spin up myself. I have basically a set of GPU workstations that I just actually treat as a local cluster. stitch and do everything on the fly. I do AI segmentation. I do mitosis detection. I do cell tracking. And then I've been working on creating an analyzed data interface that then generates, can give me my quantitative outputs and then also visual outputs. And then we have to think about the provenance in it. And so the microscope itself has some info, acquisition info, microscope info, but we really need more also about the sample info, the experimental info, what's your experiment setup, especially if you have multiple wells, and then as we're doing the processing, so I do log, let's say all the data transfers is all logged, but then also we want to say if I do a stitching algorithm, what are the algorithm that I used, did I use a fast forward, a fast Fourier transform? Did I use, this other method or, and also for the inferencing, you have to think about things like how much did you break up your tiles and, which model did you use? Did you break up your thing into 512 by 512 tiles? How much overlap do you use between each image tile that's fed in? Or did you use 1024 and so on, like for your inferencing? And then also for the tracking, like what are the cost functions that I use in the parameters? So I think the reason why I say this is because, I had a postdoc who was working on this and, writing this up. But then if I go to his, Python code, I don't know which version of Python code he used to, do this. And so I lost all the parameters that he used for this data set. What is the one that we want to use? What is the model that he used? And a lot of times you just say it's in the code and I'm like, so it's like, can we output it as a JSON instead or have it somewhere? And so what I've been working on is actually embedding it in the metadata in the actual image itself. And so every image you look at, and on the other thing I also want to say is that when you stitch, most of the stitching algorithms you do, the output image already lost all the metadata that might have been embedded in the header. And so there's also a loss of that provenance and you lose everything going up to that point. You try to log everything before, but do you know which images you truly use? And the scale of data I'm working on is each experiment, it's up to 30 terabytes. And so that's not something that you can just kind of go back by hand and, trace back to what you want. And so when you're working with big data, the automation, the metadata, everything is really crucial in order to keep track of what's going on, what's happened, what the data truly is. And so one way I've been kind of approaching this is actually creating a user interface. So I actually wrote my own desktop app to actually kind of create a user interface to input some of this, to link the sample and experimental info, and then actually execute all the Python and like cluster scripts and so on. So and this is kind of like what my script integration and tool data workflow looks like. So essentially I have an Electron-based app that's, written HTML, CSS, and then, uses Node.js to kind of do like a desktop app interface. But the nice thing about it is that it can also execute Python scripts and it can do command line runs. So I can use WSL, which is Windows Subsystem for Linux, and use it to actually run Ansible playbooks and run a Linux, like a little small Linux cluster that I set up using Dask. And then from there, I actually do on-the-fly inferencing. That's a Python script. Then I have a Python and I have a Flask server set up that allows me to also interface with the output data that I get. And I've been kind of creating that to have a video stream. So I can actually video stream the data. Because if you're actually analyzing a process for, 72 hours, 120 hours, how do you know something hasn't gone wrong and how do you trace back that data? So I was trying to think, what is the easiest way to look at this? How do you monitor this process? You just get a graphical output, like a graph? Actually, so I was thinking instead, maybe you can just go back and look at the video data output. And so the stitched images all then actually show up as like a kind of a video stream. And then you can look at each well to see what is the output. And you can look at, let's say the last 4 hours of the cell growth, did they look okay, at least visually? And then having the quantitative outputs is also important. I'm still working on that though. That's still in development. But I want to come back to thinking about metadata automation and where things really go wrong. And I think this is actually a real case scenario. This is actually my system here on the left, and I just have like this box imager there. But the thing is that, it's a bit dated. This one at least can interact with my, robotic arm. And so it's easy to kind of transfer a plate automatically into there and image it. I'm upgrading the microscope. So I have to think about what's going to happen to the AI model that I have. Will it work on that new microscope? And this was an issue that I had a discussion with basically a manufacturer that was building an automated system that's image based for doing these kinds of things as well. And you know what? They upgraded their microscope. They have a nicer one, but then their AI doesn't perform anymore. They tried transfer learning, it doesn't work. So the question is, it didn't really increase the accuracy that much. So is there maybe information in the metadata? Can we develop a way to kind of characterize what is the important things to pull out that can actually create that comparability and maybe enable the AI transferability? And so one of the experiments I'm doing right now is that I actually took just a plate of stem cells that I grew, and then I imaged them on like 5 different systems, all in phase contrast, but you get 5 very different images output from it. And so we're testing how can we do the transfer learning, how much accuracy does it do by doing transfer learning, and then also test what are the features in the image itself that the AI might be sensitive to between different instruments. There are some ways to kind of look at some features of that. And then really what I want to do is eventually, and part of what I do work with my collaborators, can we develop and incorporate possible reference materials to calibrate the AI model itself? And so that's another thing, and that comes down to like reference material, but that's a hard question, but it is something that, I probably will spend a while kind of working on, and maybe think about. And so, uh, I do have more things, but I want to kind of open up discussion. So I'm going to show you like this is so I do now have a postdoc that does a lot of she's a computer programmer, so she does a lot of the on-the-fly processing for the AI. So she's been helping me implement that, and as well as some of the metadata kind of embedding in the image headers. And then Ramila's my staff scientist that does the automation engineering. And then we have a bunch of collaborators to kind of build this whole digital twin system. So I can stop there. I do have a little bit more talking about user interfaces. Maybe I can show one thing. This is actually an example of an output I'll quickly show is that this is like just compiled from like 6 different wells. And then, talking about all the issues is that, there's a lot of data. So a lot of data is a lot of problems. And I think how do we approach this is that, try to create a structured approach for storage and to organize the data, process to analyze the data as it's generated. I probably should have shown this before, but, I wasn't sure how much time I'd have. And then also created tools to kind of generate compressed videos and other kinds of outputs. And then also the video streaming. And so one thing I want to talk about is how do you build a name? I spend a lot of time thinking about just even how to name an experiment. How do you approach this? So I actually was thinking about if you use basically like build the variables that go into the experiment. Can you actually then just, using drop downs that you can add on to them or just, enter manually and then have like a kind of shortened but still actually human readable output. I think people focus a lot on machine readable and just like, but then it's not human readable. So then you kind of take you lost one level of connection in that loop. And I think, it's a balance, do you want to generate a super long name or, do you want it also to be.
Yeah, I was just gonna say it's like it ends up being like this massively long name, and I keep
Yes.
appending things on the end, but it's so important to keep that
It is.
information.
So I think one thing that I do is I store what the full names are of the variables. So basically just select, what's the cell line, what's the media type, what's the dye, other types of variable if I'm doing flow experiments, flow as in like continuous flow, or, because I have a whole microfluidic setup that I use to kind of do automated, feeding and stuff. And then like have a shortened name for the generated output. And then I append that name into every file basically. And I also enter in like what's the dish and what's the variables in each well because you can have a plate readout on one side but you kind of lost that link because you always have to refer back to a protocol to see what happened in this exact well that I'm looking at in this image. So I actually made it so that we embed what's the actual variables in the image itself in the header. So what were the treatments that happened in this? Dyes and so on. I also do like different timings on different imaging. So I'll image in phase every 2 minutes, but I might do intermittent fluorescence as well. And, these are things that I kind of had to set up. And then I actually made it so that it automatically runs my Python script for the data transfer, and you get an output and everything, and it's also logged. And then I also have a Ansible deployment from the actual like desktop app. And so that allows you to actually do the on-the-fly processing, which is, focus finding, stitching, inferencing, and cell tracking. But, I'll go through this very quickly, but basically I had to think about how do you name everything? What is the structured approach? How do you append and shorten the names? What is needed at each step? And then how do you organize all the folders? And then also thinking about what metadata to pass on and how do you organize the metadata? Let's say you have a stitched image. Do I have a tile of, a 10 by 10 tile? What metadata do you need? There's the static metadata, and then there's also all the images that are composed within it. And so you kind of have to embed maybe what's dynamic or what's changed in each one. And so it's just kind of thinking like, the green is just the appended metadata at each step, kind of that's in the image header itself. And so it's kind of like, these are the things that I think about a lot. I spend a lot of time discussing, and it's not just, sitting down and trying to write a glossary or whatever, but it's thinking about how to really implement it and embed it into like a whole system. And the last thing I was going to talk about is like my live stream. So, these are things I just kind of wrote very quickly. They're not super well developed. They're not like nice, like tools that I would put out there just yet. But, now that I have like, a program, a computer scientist, like she can help me kind of really turn this into nicer stuff that maybe I can put out there. But basically what I do is read the metadata from each image, and then I can say what's the, conditions, and then it'll actually like play the video as well. I think, yeah, I mean, I could stop here. I think I just had what our AI outputs look like, monitoring long term, like, and then spatial relationships that we kind of do. So this is what I'm thinking about, like, what are the metrics that we generate? I just kind of wanted to show that briefly, like, how can you get that deeper understanding, not just a distribution, but we can also do spatial relationships. And so you can really analyze beyond just purely morphology or texture, but really looking at the individual cells in a population. And so I think with that, I can stop there.
Excellent. Wow, Anthony, thank you so much for sharing and presenting today. What is striking to me is that when we are in academic labs, we forget that ultimately we would like our research to become drugs and therapy. And now you're presenting to us, what are the challenges that drug and therapy developers are facing. And now if they're getting into bioimaging to monitor the bioprocess, this is highly relevant, I think, to what we're doing. And it's important that we keep that in mind, is that we're all ultimately working for for humanity.
Yes,
right?
yes.
So that's, for me, is my overall take-home message. So now I'm wondering if there are people who want to ask questions or make comment to Anthony. Perhaps the people that put
There's
comments.
a couple comments in the chat.
This
Carolyn said, I am also really interested in knowing how to validate imaging AI models. It seems like the Wild West right now, with some people going too far and others not validating at all. Yes.
Yeah, so that's a great point. So you can check out at least what we tried to do in our paper. One thing was, there's something that they use a lot in industry, like manufacturing. They call it fit for purpose. So it really depends on what your output is and how you want to evaluate. So when I'm evaluating, when I'm segmenting the cell nucleus, but I'm not necessarily trying to measure the morphology of the nucleus. And so, some people use different metrics. They use a metric that kind of measures how well the overlap between the fluorescent outline and the, AI outline is. But those don't always match. Also, there's a timing difference just slightly when you image from phase to fluorescence. And, there's a bit of registration, so it might throw off, that metric. But instead, we were using centroids and then counting centroids and then do like false negative, false positive. And so, and then we could do what they call an F1 score. So, I mean, there's a lot of papers from, I think, Ann Carpenter, she does a lot on like how to evaluate, and we were kind of using some of those, we were looking at other ways. So it really depends on what the output is, and how you evaluate it. The other thing that we did was, we developed the model on one set of data. And then what we did was test it on more biological replicates. And then we tested it on different cell lines. And then we also tested it on those biological replicates over time. And so we can really get a characterization of what's the accuracy of the model over time. Are there anything that throw off the model? Is there certain periods of time or states of the cells that are kind of causing, like loss of accuracy? And then also, how much variability is there from time to time? I did test 2 microscopes, but the problem is my second microscope system was actually relatively identical to my first one. So I had like these, and so my accuracy was, I couldn't even really detect a difference even on the same cells. It was like my accuracy was like 95 versus 94.9%, Yeah, so it was just like, it was a very very small difference. But this is why I'm actually interested in doing this where I'm testing on completely different systems. So it's like I have a Zeiss and a Nikon, and a biotech, a biotech and what else do I have? Like there's like another 2 systems now, yeah, that I had available that could do phase like right then. And so I want to really see how much of difference that we get. And this is actually more recent experimental setups that I've been doing. So we haven't gotten to the point of really getting the model outputs yet.
Can I just ask one more question? And I've got to run because I got a hard stop at 9 o'clock. So most of the instrument companies are kind of like sticking to this 16-bit, 8-bit system, right? I'm a histologist, by the way. I'm not necessarily a computer scientist, right? That's where I'm coming from. But Akoya seem to be changing all the rules, and they seem to be doing whatever they want, and they are the sort of Wild West out there, and they're normalizing things, and they're not giving us data on how they're doing it. Is there, like, have you noticed any other microscope companies out there that are doing things completely out of the box? And what suggestions do you have to try and bring us all into a fold where we understand what everybody's doing?
So I didn't do this, but my group had done this where they were looking at, normally when you're doing a processing pipeline, you might just limit everything to an 8-bit image. You scale it to an 8-bit and then do the processing. The thing is that if you go from a 14-bit versus a 12-bit versus 16-bit, that scaling is not like linear. It does not work very well.
Yeah.
It really throws off everything. And that's actually one of the things that I found out working with this other company and talking about when they upgraded their microscope for their automated system is that one had a CCD, the other system had a CMOS, and then it was also 2 different bit depths. And then they were trying to use the same kind of, they were basically the same kind of scaling down to 8-bit. And so, like those things are already throwing off, I think, a lot of things.
Yeah.
You might have differences in noise readout, might have differences in, just that scaling when you do that. That might throw off everything. And that's actually what I'm interested in actually looking at exactly. How can you do that?
Yeah.
And maybe using it, you might need an AI tool on top of the AI to fix the AI,
I think this is the way Akoya seem to be processing their data. And they're normalizing it and doing sort of spectral unmixing, but they're not clear about how they're doing it. So I think it's a longer conversation. I'm really gunning for them. I'm gunning for them though. So I'm trying to get more sort of information behind me before I go and address their reps about what they're doing. Thank you.
No problem.
Thank you. That's very valid comment. So I'm going to give the floor now to Eduardo. You have your hands up for a while. Eduardo, please go.
Yeah, but I have a lot of questions. Maybe someone else that has also to leave early. I can go at last. Thanks.
Nobody's coming forward, Eduardo. So why don't you go ahead?
Okay, so Anthony, from the beginning of your presentation, you present a workflow with some cartoons about the master bank,
Yeah,
working bank.
yeah.
at your work, do you have a way to standardize that kind of words? For example, or they come from the lab. Let me put you this example. Maybe the work parent can be used in both cases.
So master bank is a term used, I think, as an ISO term for biobanking. So there's a master bank and a working bank. This is a common term used, I think, in manufacturing. So in ISO 276, I actually sit on working group 3 as well. Sorry, they changed things. It used to be working group 2. I think it's working group. I'm sorry, it used to be working group 3. I think it's working group 2 now, but it's biospecimens and biobanking. And so, sorry, cryopreservation and biobanking. And so there was a lot of IPSC related things in there. And so I ended up sitting on a lot of these documents, but I'm pretty sure master bank and working bank are actually the terms that they normally use.
Yeah, because if we want to standardize everything,
Yeah.
this also comes pre-metadata.
Yeah, so this is how you kind of characterize that unit operation, but there definitely is, the way that you do the metadata within it, it's basically you have to have the parental derivation, and I can even see if I have that, because I have some of our other work that, we work on developing a lot of these limb systems. And these are applied also for flow cytometry consortia and so on. And so they do put a lot of thought in that. And there's a lot of terms, but each person comes out with a different term.
Yeah. And yeah, that's kind of my that was stitching that a lot of people uses every term. So the second part is about the digital twin. There is a researcher, I don't know, I don't remember her name, in Janelia, that is doing electron microscopy, and they are trying to segment every organ and organelle and every part of their HeLa cells. So are you planning to connect in the future with something like that? Because for me, that will be the next step. You have a really good way to segment the cells. And now if you connect the research from this researcher, sorry, again, I don't remember the name, but she has all the information that is inside the cell. So you have all the periphery, and now you can add the inside in general terms, but you can have the inside. And also there's another work from the Broad Institute. Then they have some key markers for biomolecule interactions. So you can start to assemble a digital twin in a really more robust way.
That's true. So there's two things, is that you can think about the digital twin as digitizing digital representation of the cell itself, which is very complicated because there's also all the dynamics within the cell. And then there's a digital twin that kind of mimics or representation of the manufacturing process and all the tools within it. And then it evaluates the cells and makes decisions and so I can tell you at least one example that we did, and I think it's been published. So my colleague Simona and Joe is building the digital twin, is that they actually put reference beads in a slide for just a cell counter for cell viability stuff. And what they found is that, if you just do it by eye, each person is a little bit different because the focus is really sensitive for those to evaluate them. And so if they put in a reference bead that actually allows you to kind of focus, get the proper focus or the same focus, you can actually kind of bring the measurements kind of equal to each other or very similar. So it's kind of like a bit of harmonization in that way. I don't know if that's the right term to use, but that's kind of the way that you think about it, is that the digital twin, they made a digital twin that actually controls the microscope focus and controls it automatically using the focus beads, and then reads the output. And so it was kind of like this autonomous system that just controlled the instrumentation. And so when I talk about it, digital twin is more on the instrumentation side as opposed to the digital twin of the cell itself. I think work from Allen Institute is very interesting as far as trying to do the subcellular, segmentation using, just the Brightfield, I think Brightfield Z-stacks and stuff. So there's a lot of cool work there that to also look at.
Yeah, for me, it's not about if we are going to get, it's when we
Yeah.
are going to get.
Yeah.
Then what? The companies are founding a lot of AI researchers. Yeah, a lot of AI
Yeah. There's
research,
already
and
a lot of it
then
going on in manufacturing. Yeah.
So I'm
and
very happy that Anthony, you're on the ISO committees, and Eduardo, you should really consider joining the ISO committees. I know that in Canada, it's been hell for me, so I gave up. But maybe Eduardo in Mexico, you might be able to join those committees, and you would significantly contribute.
yeah. Thank you. I will come to the meetings.
All right. Are they more comments? We have some, maybe Matthias, you have a question? Or
Yeah, I only would have a comment on ISO when it comes to me. So for me now, it's clear when I'm going back to Europe, and that's why it wouldn't really make sense to join ISO from that side of the ocean.
Yes, so ISO is all by country,
Yeah.
and in Canada it's been hell. So I've actually attended ISO committee as being a guest of the American or the German delegation, but it's really important. And also, Anthony, I realized last year in horror that, for example, spatial omic equipment manufacturer are actually providing even less metadata than what microscopy manufacturer. And when I found out, my first reaction was that let's put them in jail. So I mean, it's super important that BINAQUARE, but also NIST and ISO actually all work together with the industry. It's absolutely critical.
Yeah, so that was actually for me also a scary thing. I think it was like 2 years ago. I was going to some of those spatial genomics like seminars, like they were doing workshops, and I wanted to see what are you guys doing? You know, they say they're doing super resolution imaging. What kind of imaging? What? They don't say anything. They just say they have like 20 nanometer resolution or something. Like, is it? What kind of imager is it using? How well does it work? How do you benchmark? Like, how do you know it's working properly? How it's focused? Like all these things, and it is. It's just like you don't get any of that. I even asked those details; they wouldn't give you anything. So it's like, oh, okay, so this is kind of I don't know. For me, it was kind of scary in that sense because you really don't know what's going on in those. That's why those box imagers sometimes are a real black box, and you don't know. So I do work with some of these box imagers, but not like that, you know, and some of these box imagers, if you ask, they'll give you every detail about what their camera is, what's the, you know, internal components, even the manufacturer of their, you know, their optics and so on. So like this, you know, this one box imager might be using an Olympus versus a Nikon versus a whatever. But yeah, with the spatial omics, like the spatial stuff is, yeah, they just say, oh, we're doing super resolution microscopy, and that's it. That's the limit.
Yes, so very scary, and it's good that we all work together, and we want to encourage those people to join, and that brings me back to data model, metadata, and ISO standards. So it's super critical that we keep working together.
I agree.
All right. And anybody else? We're already past the hour. I'm inviting people to raise their hand.
I mean, if anything, you can always contact me by email. So
well,
reach out.
Anthony, I'll say right away that you'll be getting an invitation to present on the Digital Research Alliance of Canada.
Oh, really?
Because I'm now on their metadata expert committee, and we have a lot of for life sciences, we have a lot of representations from the genomics people, and I'm still fighting with them to have them understand that bioimaging is a lot more complex. There was a meeting last week in Montreal, and the guy was saying, we generate 1 petabyte of data per year. And I'm like, oh my God, we can do this in 48 hours with
I know.
all these high content screening system. So, but it's actually very valid. So you'll be hearing from me
Okay.
on
Absolutely.
behalf
Yeah.
of Canada.
Thank you.
Thank you. All right. So once again, thank you so much. Big applause. And we look forward to having you all in the next AIMM meeting.
All right. Thank you for
Thank you so
sticking
much.
with me through my whole talk.
Very
so.
valid. All right. So everybody have a wonderful time between now and the next AIMM meeting. And we look forward to welcoming you again.
All right. Thank you.
Take care.
Thank you.
Thank you.
Bye-bye.
Bye.
Bye.